Julia Davis-Porada in Microbiology and Immunology will present how vaccines generate protective immunity in human tissues.
As a vaccine scientist, I certainly remember getting my first COVID-19 vaccine in December 2020, and I even took a selfie. But do any of you remember getting your COVID vaccine? Yes, but how about a flu, or chickenpox, or measles? If you don't, rest assured that your immune system does. When your body is exposed to foreign material like a virus, it rapidly responds by producing proteins called antibodies and immune cells that are maintained in your body over time as memory. This immune memory is what provides protection from infection if or when you're exposed to that same virus again. And it is this memory that a vaccine is meant to replicate. In my studies, I evaluate the ability of the novel COVID-19 mRNA vaccines to generate immune memory. But unlike most studies of vaccines in humans that only focus on blood, I look within organ tissues, which is where the majority of immune cells actually reside. So specifically, I collect tissues from human organ donors and isolate single cells from bone marrow, spleen, lung, and lymph node, in addition to blood. And by surveying the vaccine response across all of these tissues, from donors in their 20s to their 80s, I have arrived at 3 major findings. First, vaccine-induced immune memory is predominantly stored in lymph nodes, and there are certain types of immune cells that don't exist in the blood at all. Therefore, vaccine development must test on lymph nodes in addition to blood. Second, while immune memory declines in the blood over time, I found that immune memory actually increases in the spleen and is steady in other tissues. And this suggests we may need to reevaluate the timing and frequency of booster vaccines. Third, data suggests that immune memory in blood declines more rapidly in elderly patients compared to middle-aged patients, leading the CDC just last week to recommend more boosters in patients over 65. However, in the tissues, I found no correlation between the quantity of memory and patient age. Together, these findings demonstrate that immune memory in blood does not represent immune memory in tissues. In order to understand the full breadth and durability of an immune response to a vaccine across the entire population, we must study tissues. I hope that this work is the first step toward the creation of new vaccine testing programs that use lymph node samples and the beginning of the inclusion of tissue research in determining vaccine guidelines. That way, you will all remember your vaccines, even if you don't take a selfie. Thank you.